Hi, and good afternoon, everyone. My name is Dr. Charles Chiu. I'm a virologist and a microbiologist at University of California, San Francisco. And I'll be talking to you today about viruses and acute flaccid myelitis, or AFM. So I just want to briefly review the terminology before we get into the viruses. So acute flaccid paralysis is defined as a clinical syndrome of acute onset focal weakness in any part of the body. And it is the epidemiologic case definition that traditionally has been used for poliovirus surveillance. It does include a wide variety of both infectious and non-infectious etiologies. And prior to, say, 2014, pretty much all of these clinical cases, regardless of whether they were caused by a specific viral infection or associated with a specific viral infection or due to a non-infectious etiology, were called AFP, or acute flaccid paralysis. And in fact, poliomyelitis, which is the poliomyelitis virus, or the poliovirus, it actually it originally did not refer to the virus, but it really referred to the pathologic syndrome that was caused by the virus, which was a disease of spinal cord gray matter associated with inflammation. And the virus itself that was discovered to be the cause of this poliomyelitis was subsequently named after the clinical syndrome. And then as additional infectious causes have been identified, various terms have been used to describe this. They include poliomyelitis, polio-like paralysis, acute flaccid paralysis, and acute flaccid myelitis. So when we think of the case definition of acute flaccid myelitis, and this has also been, Kevin also earlier discussed this, is that you have to meet the clinical criteria, which is acute onset flaccid limb weakness. And in addition to that, there's imaging criteria, which include an MRI scan, magnetic resonance image, showing a spinal cord lesion that's largely restricted to the gray matter and spanning one or more spinal segments, or having pleocytosis, which is white blood cells in the spinal fluid. So when we think of AFM, we typically think of the viruses that are associated with this condition. And we can really think of AFM as a subset of acute flaccid paralysis. So going through the viruses, so originally, probably the prototype cause of acute flaccid myelitis is polio, or poliomyelitis. This is a disease that's spread by fecal-oral transmission of poliovirus. What's also interesting is that fewer than 1% of those who are infected actually get the paralytic disease. And you'll see later on that there's some ties with perhaps with enterovirus D68, where a very small fraction of children who get infected end up getting paralytic symptoms. With poliovirus, it began with a viral upper respiratory infection, typically begins with a fever and a sore throat, that then can persist and progress into a flaccid weakness. Usually in spinal fluid, you can see a mild lymphocytic elevation of white blood cell count, pleocytosis, as well as mildly elevated protein. You can detect it in the stool of the virus for weeks to months. The virus can be shed in stool, but it can be very rarely identified in spinal fluid. In fact, less than 1% of the time, you can actually detect poliovirus in children who have poliomyelitis. And this will be a theme or a trend which really spans not only poliovirus, but other viruses, including enterovirus D68. Another virus that's been linked to AFM, or acute flaccid myelitis, is Enterovirus A71. Enterovirus is a very broad group of viruses that they're causes of the common cold, and they cause a wide variety of different types of infections. They are spread fecal-orally through contaminated food or water. With Enterovirus A71, what's interesting is that you get a syndrome called hand, foot, and mouth disease, where children develop lesions and in the various areas of their body, their mouth, hand, and foot. And it's also associated, hand, foot, and mouth disease is not only Enterovirus A71, but it's also associated with other enteroviruses. What you can see on MRI, magnetic resonance imaging, is our brainstem and anterior spinal cord lesions. And it does cause a flaccid limb weakness, which clinically is diagnosed as AFM, or acute flaccid myelitis. Unlike, though, enterovirus D68 AFM, which I'll talk about shortly, most children who develop enterovirus A71 associated AFM recover completely from their disease. And I think this is what highlights the importance of being able to diagnose a virus that is associated with any child's given condition, because some causes of AFM, viral causes, appear to have a better prognosis than others. Similar to poliovirus, it can be detected in stool, and in skin lesions, but very rarely in spinal fluid. And since the 1970s, there have been periodic epidemics of enterovirus A71. And in fact, as Dr. Ruth just commented, in 2018, there was a local epidemic in Colorado of enterovirus A71, which explains some of the cases of enterovirus A71 associated myelitis in 2018. Another virus that's been linked to AFM is West Nile virus. This is an arbovirus, meaning that it's spread by vectors such as mosquitoes. It's a flavivirus. It's transmitted by the mosquito. It arrived in North America in 1999 and now is endemic. We see seasonal epidemics occasionally of West Nile virus. Similar to other cases, you can have 80%, similar to other viruses, 80% of individuals who develop West Nile virus infection are asymptomatic, have no symptoms whatsoever. Roughly 20% have perhaps a flu-like illness, and then less than 1% develop this neuroinvasive disease that can mimic AFM caused by any other virus. Typically, the spinal fluid is also negative by doing sensitive testing, looking for the virus at the time of clinical presentation. So what we have to do to diagnose West Nile virus often is we have to detect antibodies that were made by the patient in response to the infection. And this becomes very important when I talk shortly about diagnosing enterovirus D68 associated AFM. I also want to mention that there's another virus. It's not as well known perhaps as these other viruses. It's called Powassin Virus. This is a virus that's spread by the same Ixodes tick that carries the bacterium that causes Lyme disease, Borrelia burgdorferi. It can present, infection with Powassin virus can present similarly to these other viruses with upper respiratory, signs of an upper respiratory viral infection, such as fever, headache, common cold symptoms, but it can rapidly progress into developing a neurologic disease. And in fact, there have been case reports of essentially acute fussy myelitis, meaning that the Powassin virus infection met all of the clinical criteria for AFM. And again, you see the same theme here in that when we try to look for the virus in the spinal fluid, the PCR test is often negative, meaning that we have to rely on antibody testing or some indirect method of diagnosing acute infection. So I want to end by talking specifically about enterovirus D68. From 2012 to 2014, there was an outbreak of poliomyelitis-like cases in the United States. This was initially highlighted by Dr. Carol Glazier, my collaborator, who I've been working with on the California Encephalitis Project. She, as part of her surveillance with the California Department of Public Health, had received reports of unexplained paralysis, requesting poliovirus testing beginning in fall of 2012. The California Department of Public Health then instituted statewide surveillance, and this was when the syndrome of AFM began to be characterized. And then 2 years later, with this biennial cycle in 2014, there was an unusual cluster of similar paralysis cases in Colorado, which were managed by and really heralded by kind of the work by Dr. Kevin Masakar. So this is an identical slide to the one presented by Dr. Ruth from the CDC, showing that we have these biennial outbreaks, and we don't really know what's going to happen for this year with respect to acute fussy myelitis. But the key question is, what is actually causing, the recent cases, these biennial outbreaks of AFM? One thing is, as I said before, for many of these viruses, we're unable to identify the virus actually in spinal fluid. We've even applied a method called metagenomic next-generation sequencing, which looks at spinal fluid and looks for any virus. And I can tell you that we've now looked at over 100 spinal fluids from children with AFM, and we haven't detected any virus, including enterovirus D68, directly in spinal fluid. In addition to that, typically the testing didn't show poliovirus positivity. There was no evidence of West Nile virus or Powassin virus or other arboviruses. And really the prime candidate for this in 2014 and 2016 was the fact that we did detect enterovirus D68, but we had to survey non-sterile sites, respiratory secretions and stool, and we couldn't find it in spinal fluid. And this, to some extent, made it very difficult for us to assign causality to enterovirus D68 because we can't find it exactly at the site of the presumptive infection. So a few words about enterovirus D68. This was a virus that was initially discovered in 1962 in respiratory specimens from California children. It's an interesting enterovirus. Most enteroviruses kind of grow at body temperature. This one appears to grow at 33 degrees. So it behaves more like a common cold rhinovirus than it does like an enterovirus. And it appears to be transmitted by respiratory droplets and causes primarily respiratory disease. But for many years, in fact, for decades, it's really been thought to only cause the common cold. And it was only until 2012, 2014, where we started to see, where we had a widespread outbreak of enterovirus D68 respiratory disease. In fact, in 2014, there were over 1,100 confirmed infections in the United States, of which a very small proportion of children who were infected developed AFM. So the key question is, is this really a cause of AFM? And I'm going to present a lot of data in just 2 or 3 slides. But the general idea is that researchers have been working now for many years on trying to essentially prove, is enterovirus D68 causing AFM, at least in these biennial outbreaks. There is some very intriguing epidemiologic data. If you look at the California longitudinal surveillance, the AFM incidence increased more than 5 times during the 2014 outbreak than it did in previous years. And this happened to be significant, quite significant. When we do sampling, and this is done by we, I mean the CDC and other public health agencies, do sampling for respiratory secretions from children who have AFM, we find typically enteroviruses, D68 in 2014 and 2016. And then we, as I mentioned before, we also saw A71 in 2018. So this was an epidemiologic link to AFM. Some other data is data that we obtained from genomic sequencing. And so what we did in this study is we looked at all of the early cases in California and Colorado of AFM, and essentially all in children, or nearly all in children, from 2012 to 2014. And what we found were some very interesting findings. One was that we found that all of the cases actually that were associated with AFM, they basically were all part of this clade B1 strain of enterovirus D68. And that was, in fact, the clade B1 strain was, in fact, the outbreak strain that was causing the vast majority of all of the respiratory cases of enterovirus infection. The cases with enterovirus D68 detected in respiratory secretions also had an average 5-day prodrome, and we were more likely to detect it in respiratory secretions if we were able to get the sample within 7 days after the onset of symptoms. Past 7 days, then we were unable to recover to detect the virus in respiratory secretions. And as I said before, we've been unable to detect the virus in spinal fluid. I should also mention work done by Dr. Ken Tyler and colleagues, where they essentially inoculated mice with enterovirus D68, and you can actually deliver the virus either intravenously directly into the muscle, or you can even have the mouse inhale the virus, and it can cause a clinical syndrome that essentially resembles in the mice that resembles AFM. They get hind limb paralysis, unilateral, in some cases involving multiple limbs. What's also interesting is that depending on which side of the mouse you actually inoculate the virus, you can then induce the muscle paralysis on that side. And I just want to finally end by talking about indirect ways that we can implicate enteroviruses in AFM. So this was work done by my colleague, Dr. Michael Wilson at UCSF. He developed essentially a way to survey for antibodies to all viruses directly from clinical samples like spinal fluid. And so this was a study where in a blinded fashion, they took spinal fluid from children who had AFM, and they compared it to children who did not have AFM, who had the other neurological diseases. And they found that there was a very high proportion of cases in children with AFM had specific antibodies to enteroviruses. Now, this particular assay that they're using doesn't distinguish between D68 or enterovirus A71, but it is quite convincing that there appears to be an enterovirus-specific response. I also want to just highlight some of the work that we're currently doing. We're actually looking at it from a different point of view. Instead of using antibodies, we're looking at what we call the human host response. We're actually looking at the patterns of genes that are being expressed by children during the infection. And by doing so, by looking at spinal fluid from children, for instance, with enterovirus A71 AFM and D68 AFM, we can identify and be able to, in principle, diagnose what virus they actually have without being able to detect the virus directly from spinal fluid. And it's our hope that we would have this, a test like this, a host response-based test, perhaps available in time for surveillance, in time for the next outbreak of enterovirus D68 associated, enterovirus-associated AFM. And I want to end with one last slide, and this is a sort of a segue to the next talk, is that there's been a thought of like SARS, coronavirus 2, and whether or not has this been associated with neurological illness. This was actually a case from just 2 weeks ago, a 14-year-old boy from the San Francisco Bay Area who presented with seizures in mid-March. Eventually, so had neurological symptoms. Eventually, we were able to discover that he had been infected with SARS coronavirus 2. And we actually tested his spinal fluid, and it turned out that he had an autoimmune disease, an NMDA receptor antibody encephalitis, which was his diagnosis. And the presumptive diagnosis at this point is that he had developed an autoimmune encephalitis that was triggered by COVID-19, triggered by SARS coronavirus 2. So it sort of highlights the fact that multiple viruses can cause an identical clinical syndrome, which really underscores the importance of being able to accurately diagnose the viral infection. So I'll stop there and then segue into talks from Dr. Grenfell and Dr. Park regarding the impact of the COVID-19 pandemic on enterovirus circulation and AFM. Thank you.